And thank you, Varijit, for inviting me for this very interesting meeting where I'm learning lots of new things and meeting lots of new people. Um, I will talk something which is perhaps not directly in the mainstream of computational biology, but it's in the larger scheme of mathematical modeling of biological systems. Um, um, this is work that has been done in collaboration with PhD students, Parth Pratim Pandey, Pooja Sharma, Harshan Singh, and Shagun Nagpal Sethi, different times. Here's the outline of the talk. I will describe some system-level phenomena in bacteria, in particular, exponential growth of single cells in steady-state cultures. Most bacteria, as single cells, their size grows exponentially with time until they divide. And I'll talk about that. Also, most of the constituents inside the cells, they increase in a balanced way, in that they increase with their ratios sort of maintained over time, broadly speaking. This, I would like to, there are other phenomena which I will not be able to talk, I've just mentioned them here, but these models also, and their extensions also talk about, discuss those phenomena, which are in the third bullet. The question that I will be interested in is how can we understand the first two phenomena, exponential growth and balanced growth. And there is actually a mathematical puzzle underlying these phenomena, and I'd like to explain what that puzzle is. And it turns out that there is a certain mathematical structure that naturally arises in the dynamics of bacteria, which answers that puzzle. Okay, and so I'd like to start with this 1962 result of Schechter et al., in which on the x-axis is plotted time, on the y-axis is the logarithm of the length of a bacterial cell in a culture. And there are different cells which have been, each line corresponds to a distinct cell. And the y-axis, is sort of somewhat arbitrary in terms of its intercept. Different, they've been placed in different points to distinguish them. But what is interesting is that the length of the cell, which is a surrogate for the volume of the cell, because it turns out that E. coli, in a fixed medium, they grow with their rod-like cells, but the width of the rod remains constant, and they only elongate in length, and therefore the length is a surrogate for the volume itself, for the cell volume. And they measured this, and because it is a straight line for all these cells on a log linear plot, that means that cells are growing exponentially with time. Okay, the size is growing exponentially with time. These things have been measured in much more detail in recent years. What I'm showing you here in this slide, so that the first red plot on the left is a series, is a cell lineage which has been studied across different generations, and the size of the cell length, rises with time. Time is on the x-axis again, and then when the cell divides, then the thing abruptly falls, and one of the daughters is tracked. So these traces are cells growing and dividing, and you're measuring the length of one lineage of cells, starting from some mother cell, then one of its daughters, then another daughter, and so on, as it divides into two daughters. Okay, and so that, if you plot, if you look at the third picture, which is, I should use this. This one, where you're looking at a particular trajectory of a single cell from growth to division, that is very well approximated by an exponential curve. And, this happens in a number of different bacteria. Similar kind of things happen. And, this curve is about not the size of the bacterium, but the amount of a particular protein in the bacterium. And it turns out that, these are also growing. The amount of different chemicals in the bacterium are also growing exponentially as the bacterium is growing. Okay, its size is growing exponentially, and that is basically responsible ultimately for the concentration of different things to reach a constant. Because if the chemical populations are growing in proportion to the size, then the populations will become constant, and that is what is the steady state of the bacterial culture of a cell in a bacterial culture. Now, I want to now explain why this observation is actually a puzzle, and the reason. So let's try to model a bacterial cell as a dynamical system, and one approximation, one model which makes a lot of approximations, but is a model that is a poor man's model. That's something that we can hope to do at this time, with certain amount of coarse graining and so on, is that let's say that the cell consists of n chemicals, okay, whose populations are going to be given by this capital X1, capital X2, and that these are functions of time. And so that basically the amount of all the chemicals, n of them, 1,000, a few thousand chemicals. If you have, once you know the populations of all of them, then you know the state of the cell at any particular time. That's our state, okay? And you can specify a deterministic dynamics in the form of a first-order dynamical system. dxi by dt, or xi dot, is some function of the existing populations. And, these functions, so there are n such equations, and the functions on the right-hand side, the ith function, is telling you how the ith chemical population is changing with time, and it has the contribution of all the reactions that are producing that chemical, the reactions that are consuming that chemical, and everything that impacts the population of that chemical, transport into the cell of a particular chemical, those that can be transported, excretion, and so on. Right. So that so it contains all the reactions, all the interactions are the complexity of the cell is inside these functions. And these functions are in general highly nonlinear functions of the variables xi, because chemical dynamics is nonlinear. Okay, and that is the source of the puzzle. That how is it that in spite of this obvious non-linearity, you get an exponential behavior in a steady state of the size of the cell, and each of these chemical trajectories, which is the hallmark of a linear system. Okay, so let me just make a brief digression to a one-dimensional dynamical system. dx by dt is some constant r into x to the power alpha. Okay. So this is just a single system, and alpha is equal to 1, of course, is a linear system, and alpha less than 1, alpha greater than 1 are examples of non-linear systems. Okay, and you have this is now a simple differential equation, and you can solve this differential equation, of course, in elementary calculus for any value of alpha. And here I've written down the three solutions for alpha is equal to half, for alpha is equal to 1, and for alpha greater than 1. So for alpha is equal to 1, you have, of course, the well-known exponential solution. X naught is the value of x at time t is equal to 0, and then e to the rt, where r is this constant. And that's the solution that you have for a linear system. But the moment you have any nonlinearity, you don't have an exponential solution. Okay, so here you can see that there is a power law behavior in time. X of t depends upon t, rt plus something, initial thing, and then square, and so on. And whereas here, so that grows slower than exponential. This power law behavior is slower than exponential. Whereas if alpha is greater than 1, then you have a growth which is faster than exponential, supra-exponential growth, which in fact you can see that it has a finite time singularity. That at a finite time, which is the reciprocal of Rx naught, this denominator goes to 0 and the population, the variable will diverge at a finite time. So that's what you expect in any nonlinear system, even in one dimension. Okay, so we of course have a nonlinear system, and why then do we end up with exponential growth? Okay. So what happens if you go beyond one dimension? Okay. So here is an example of a linear system, but n-dimensional, right? So there are n variables xi, and dxi by dt is a linear function of the x's, where the a is a constant matrix. Okay, aij's, they form a constant matrix, so this is an example of a linear function in n dimensions, a linear dynamical system. So one can write it in matrix form, and here it turns out that there are transients in which the behavior is not exponential, but eventually the system gets attracted to a trajectory in which all the variables grow together exponentially. And this trajectory actually, these are the, so let lambda 1 be the largest eigenvalue of A. A is a matrix. Corresponding to that, x lambda 1 is the eigenvector. And the eigenvector of A corresponding to its largest eigenvalue is basically the attractor of all trajectories. Trajectories, starting out with any initial condition, eventually all trajectories will converge and they will start growing along the direction. The eigenvector defines a direction in your n-dimensional phase space. Okay, and all trajectories will converge to that and start growing along that. For long times you will reach that. So effectively, in a steady state, you have recovered exponential trajectories. So even in n dimensions, there is no problem in understanding why linear trajectories arise why exponential trajectories arise for a linear system. Okay, and of course, when once it has reached that attractor, then you can see that all these, because it's an eigenvector, then all xi grow with the same rate, e to the lambda 1t, and xi of lambda 1. Okay, that's the the eigenvector. So the components of the eigenvector, right? They are the relative populations of all the species. If these xi are populations of species which are obeying this linear equation, then that's then the ratios of the populations are preserved, and that's what we want. That's what we would call balanced growth. But we have nonlinear systems, and I will give you an example. So there are lots of chemicals transported in, converted through these chemical reactions, and proteins are produced, and then there's transcription, translation happening, and so on. One can write down a complicated model, but what I will do is explain this puzzle and its resolution in the context of one model, which is a coarse-grained model of a bacterial cell, which I will call the PTR model, because there are three variables. P, which stands for precursor molecules. Let's call them amino acids. Okay, these are amino acids which are precursors to make proteins. T stands for, let me see if I've explained it anywhere. Yes. Right. Okay. So P, so T are the transporter molecules. T stands for not only the transporters but also the enzymes which are making these precursor molecules. So the T molecules bring in the food and also metabolize it to make the precursor molecules. And these precursor molecules are then assembled. Amino acids are assembled to form the T molecules and also to form the ribosomal proteins. So the third variable is the number of ribosomes in the cell. Okay, and ribosomes are catalyzing the production of the enzymes the transporters and themselves, they are assembling these P molecules, essentially, if you like, into the complicated macromolecules of the cell. So this is a three-variable, coarse-grained model of a bacterial cell. P x1 and T x2 and R x3. And so, you can write this model down in terms of three differential equations. Okay. And here is, given the roles that each of these things play, you can write down equations like this for the concentrations. Okay. So my notation is that if there is a box bracket, that means concentrations. If there's no box bracket, then that means populations. Okay. So for the concentrations, you can write down an equation like this. So dP by dt has a term that is proportional to the concentration of T, okay, because T is what's bringing it in and producing P. So there is a term that is proportional to T, and these constants, depend upon the food source, its richness, and its the transporting efficiency of these transporter molecules, and etc. Okay, there is a negative term which is the consumption term, but in order to explain this, let me just go to the T and R equations. So T, the enzymes, are produced because there is this the P molecules are consumed, and the rate of their production of the T molecules is proportional to the concentration of P and the concentration of ribosomes, because ribosomes are the catalysts that are trying to produce them. This is again a simple way of treating catalysis. It's a very sort of crude coarse-grained model, and there is a constant here, which is which tells you the rate at which ribosomes, the rate at which these T molecules are being produced. Okay, it captures the efficiency of ribosomes. It captures the size of the molecule. MT is the number of amino acid monomers used, and FT is the fraction of ribosomes making T, because ribosomes are not only making T, they're also making themselves. They're also making ribosomes, and the fraction of ribosomes making ribosomes is FR, and each ribosome requires MR number of precursor molecules, and so these are the rates of their production. And when you add these things up, you have a similar rate of consumption of P, because P is basically being used up to make ribosomes and proteins. Okay. Now, this kind of a model has been used to address various experimental questions in bacterial physiology, and a model like this actually ends up explaining things called bacterial growth laws, which tell you how the ribosomal composition changes with growth rate of bacteria. So this molecule, coarse as it may sound, can be used to explain various experimental facts about bacterial cells. But here, I'm not going to focus on those facts. I'm going to focus on the fact that this is a nonlinear model. Okay, you can see the quadratic terms on the right-hand side. Okay, and we want to understand exponential growth in a model that is highly nonlinear. Okay, so what we must do is, of course, our cell is an expanding cell. The bacterial cell, its size is growing, and these are equations that one would write down in a fixed size container. If you want to write down the same equations in an expanding container, then you have to add this term, minus dV by dt divided by V into xi to each equation. So we have to add this dilution term, okay, which reduces the concentrations on for each equation. Okay, and these are the equations that you get. Okay, and these are the equations that you need to solve for the concentrations as a function of time. But notice that these equations are not complete, because I have not told you what is V dot by V. Unless I tell you what is V dot by V, I have not specified the dynamical system. Okay, these constants I would of course have to specify, and I presume that they've been specified. But even after specifying all that, unless I tell you what is V dot by V, I have not really specified the dynamical system. So in order to complete the story of specifying the dynamical system, I will look at the model in terms of populations rather than concentrations. That's what we started with. And so populations are just concentrations times V. And therefore, if you want to write down the rate of change of populations, then each population will have two factors, and its rate of change will have these two terms, and you have to add that extra term there. And if you do, well, it turns out that this extra term will precisely cancel off. It will precisely cancel off this extra term that you had added on here in the concentration equation. That term gets precisely cancelled off, and you get these simple equations for your populations. Okay, populations in an expanding volume will still satisfy these simple equations, but these are nonlinear equations. Okay, and now here is where we make a key assumption. Okay, so these equations are also not completely specified unless I tell you what V is. Okay, and we specify V to depend upon the populations. So, depending upon how much stuff there is inside the cell, the amino acid molecules, the transporters, the ribosomes, etc., the volume of the cell will depend upon that, because as this concentration, as these numbers grow, osmosis will drive water into the cell, because the concentration of water will have declined. As these numbers, if these numbers grow, the concentration of water inside the cell declines, and in order to maintain the osmotic balance, water will be pushed into the cell. So water will, the volume will be an increasing function of these things, and we assume that it is given by this kind of function can be justified based on osmosis, although the details have to be worked out. Okay, but we assume that you have a linear function. And in general, you can generalize this slightly, but okay, so we assume this. Okay. Now, once you've done that, then let's take an example. You take, let's say that all these constants are the same, and so you have some little constant V plus P plus T plus R. Okay. Once you've specified the volume this way, and you've specified this constant V, now the model is fully specified, because the right-hand side is completely specified as a function of my unknown variables. All the functions are clearly specified, and now and then just you have to know the constant. And then in terms of the parameters, the function is completely specified. But notice that V dot by V, which is also called the growth rate of the cell, is not given in terms of the concentrations. Why? Because V dot by V, is simply, since V is this expression, V dot is simply this, but P dot, T dot, and R dot I know from these equations, and when I substitute these equations in this expression for V dot by V, they get converted into concentrations. Okay. So I have expressed this unknown, this quantity, which was not known in this equation, in my concentration dynamics, which was not fully specified, also gets fully specified now, because V dot by V gets specified as a function of the concentrations. So notice that there is a non-trivial function of the concentrations that gets added to the dynamics, because you made the assumption that the volume is no longer constant but depends upon the populations. And that assumption, therefore, makes this model more complicated, because those things are there. But it doesn't matter. Now it's a non-linear dynamical system with concentrations. Okay. Now, you see, okay. So the concentration dynamics, it is very well known. You can have nonlinear dynamics of concentrations, and many of these dynamics very often lead to some fixed point. Because there's some stable fixed point in the dynamics, and everything gets attracted. All the concentrations get attracted there. And let us say there is some stable fixed point in which the concentrations get fixed. Okay. They become time-independent constants there, right? So if you have such an attractor, and in fact this system, which I've told you, right, it does have that kind of an attractor. Okay, so it does have, in fact, one can show that it is a agreeable attractor. doesn't matter what initial conditions you start from, you will converge to that fixed point. Okay. So once you've converged to that fixed point, then these are all zero by definition because it's a fixed point. All the left-hand sides vanish. Okay. That's a steady state of the of concentrations. Okay. And when that is a steady state, what that means is that mu has become a constant. Okay. So P, T, and R, if they become constant, so after that mu becomes a constant. And since mu is V dot by V, if mu becomes a constant, that means V must grow exponentially. Right? So the very fact that our concentration equations has a fixed point, has an attractive fixed point, means from this equation that V must grow exponentially because V dot by V is a constant. Okay, that's precisely the equation for exponential growth. So if V goes as a constant and these populations are simply the concentrations times V, and concentrations are constant, it follows that each population is also growing exponentially with the same growth rate as V. So we have recovered in this system exponential growth of the populations, all of them balanced because the ratios are constant. Okay, growing in concert with the volume. So that's what's happened. How much time do I have? 5 minutes? Okay. Now, so, you can do simulations of the model, and that's what turns out to be the case, that here is time, and here the populations P, T, and R plotted as a function of time, starting from some arbitrary initial condition. There is a transient, and then after that, after some time, you see this is a logarithmic scale on the y-axis. So this straight line means that they are growing exponentially, and all of them are growing exponentially at the same rate because the slope is the same. Okay. And this doesn't depend on initial condition. You can start with a different initial condition. There will be a transient, and the same thing will happen. And of course, if you look at their ratios could be you could start with any different 15 orders of magnitude difference, but they will all converge to fixed ratios after a certain time. So balanced growth is a consequence in this model. And what I will do in the last few minutes is to explain to you how this has happened. What is the underlying reason why a nonlinear dynamical system is showing us such a simple behavior? And the reason is that if you look at these functions now, F1, F2, F3, which are on the right-hand side of my population dynamics, which are these functions in this example, you will notice that because of my choice of V being linear in the populations, these functions are homogeneous degree 1 functions. What that means is that if I multiply all the variables in these functions by the same constant beta, and use that as the argument of Fi, then that argument beta comes out, right? That means it's beta times Fi of x, because and then the reason for that is that this V is linear. So if V, if I rescale all the variables, V goes to beta times V, and on top you see there is a quadratic function, P goes to beta times P, R goes to beta times R, and therefore this term, though it is highly nonlinear, still goes to beta times itself. Okay, so this is what has happened, and of course this term was linear anyway, so that of course scales as beta, but this term was nonlinear. All these terms were nonlinear, but the nonlinearity is appearing in precisely such a way such that when you rescale all the quantities by a certain factor, the entire term scales by the same factor. Okay. And this is the mathematical reason why an exponential solution. So if you make an ansatz, so if I have this as my differential equation, but this function f satisfies this homogeneous degree 1 property, and I take this ansatz and substitute it in this equation, then you see on the left hand side I will get mu xi into e to the mu t. On the right hand side, I would have e to the mu t inside each of these arguments, but then using the scaling relation it will come out, and e to the mu t will cancel on both sides. Okay. So therefore, I will be left with an algebraic equation, which is a nonlinear algebraic equation because these f's are nonlinear functions. Okay, and that's a generalization of an eigenvalue equation of a matrix. I'll get this, and the exponential solution is a solution to nonlinear differential equations where these functions are homogeneous degree 1. So this is what we are ending up is with a generalization of linearity. Okay, so this is a linear differential equation. Okay, and by definition, such a function fi is manifestly homogeneous degree 1. And this one, of course, we know that the eigenvector, the eigenvalues of this A matrix and the eigenvector basically determines the exponential growth. But something like that is also happening for this nonlinear system, but except that you have this nonlinear generalization of the eigenvalue equation. Sorry, okay, nonlinear generalization of the eigenvalue equation that you have to solve, and mu is the eigenvalue. Okay, so I have shown you that in this example, I have shown you that in the example that I've talked about, this PTR example, we are going to get homogeneous degree 1 functions. But now I want to claim that you will always get homogeneous degree 1 functions. Okay, whenever you are doing mass action kinetics, but the volume is itself either a linear or a homogeneous degree 1 function of the arguments of the variables. Okay, this is going to always happen, and this is a simple proof of that. But since I've run out of time, I'm not going to dwell on this. But you can easily see that even if you had some generic equation in terms of concentrations, so in our example, G1, G2, G3 with these functions, could be any function, any nonlinear function. When you consider an expanding volume, you will add an additional term. But then, in the case of populations, you will get back to this equation. And now, you can see that if your V satisfies this property, that V of beta x is equal to beta times V of x, then these functions fi, okay, will be basically homogeneous degree 1. So it doesn't matter. This is just a particular example, but any kind of model that you write down of this nonlinear dynamics, okay, which comes from mass action kinetics, which means that it can be written in terms of concentrations like this. Okay, that's what any function of the concentrations, it will have to have this structure, and therefore it will have to have exponential solutions as solutions to the system. Okay. So this is such a generic mechanism. Notice that we didn't have to use any regulation to get exponential growth or balanced growth. Normally, it is supposed that so many chemicals are conspiring to double at the same time. When a cell has to divide into two identical daughters, right, and they have to be identical to the mother when it was born, you need to have an exact doubling of all the chemicals. Okay, and how do decentralized chemicals double together? That's a puzzle of balanced growth. Okay, normally it is supposed that there are various regulatory mechanisms inside cells, various checkpoints and so on, which ensure that everything ends up being the right amount when the cell has to divide. What we are finding here is that you don't need any such mechanism. This is going to always happen provided there is a fixed point of the concentration dynamics, and provided, of course, that the volume is a homogeneous degree 1 function. Okay, so this is a very generic mechanism, and therefore it must also have been true for protocells at the origin of life. It is not it is not a mechanism which has been sort of invented in the course of evolution. It is something that one can presume might have been a very primitive mechanism at the origin of life. And in fact, there is some work that we've done which actually uses this mechanism for protocells to understand protocell evolution. That's okay. So you can do this with many variables and so on, and you find the same result. But if you depart from homogeneous degree 1 property, that is, if your volume, for example, is not a linear function of the populations, then you don't get balanced growth. Okay. These models we've used to understand, along with stochasticity, to understand cell-to-cell variations and the adder property of cells. But that is not something that I was going to have time for. I will simply conclude here by saying that there seems to be a simplicity underlying a very complicated dynamical system. Okay, and this simplicity is something which is first phenomenologically visible. Okay, through these exponential trajectories that bacteria have, and through the balanced growth that bacteria have, and this observed phenomenon basically leads us to understand what is the underlying mathematical simplicity behind these models. And you can, of course, make models which are very complicated. You can, I've presented a 3-variable model, but you can, on this theme, put in all the complications of, the few thousand variables inside cells, and this property is robust and it will be preserved. Okay. So that's what I have to say. And just last work, that this structure, this nonlinear generalization of the eigenvalue equation, is something that I have not seen studied mathematically. Okay, so this is matrices and their eigenvalues and eigenvectors, of course, are an old subject, but the nonlinear generalization of that is something that might be of interest to mathematicians. Okay, as a structure to investigate the properties of. So it is of interest, of course, to people understanding bacteria, but also perhaps to mathematicians. Thank you.
Questions? Fantastic presentation. And first of all, a disclaimer, I am not from this domain, so I could not understand the part of it. But a very nice question, this PTR theory. So you're saying this amino acids will make the transporters, they will make the ribosome, right? So they are getting consumed, and as a reason of this, the volume is changing.
Yes. So the volume is changing because there is an overall change in the number of molecules inside the cell. This is part of it. The fact that they are getting consumed is part of it. But also, of course, food molecules are coming in. Okay, and that's where the number of molecules inside the cell is increasing. When these molecules, P molecules, are getting used up into making T molecules and R molecules, if you like, the number of molecules is actually getting reduced inside the cell, because the T molecules, each molecule uses up 300 or 400 amino acids, and ribosomes consume a few thousand amino acids. But all that stuff is eventually coming in, the first term in those equations, which is where, so look at it. This term, P dot, is KPT. Right? This is a positive term. This is where the transporters are getting your stuff in, and this Kp hides a lot of complexity. It's a constant which hides all the complexity of what's the medium, what's the concentration of food in the medium, and what is coming in, and so on, right? But that's the term that is the culprit, which is causing an overall increase in the number of molecules, and hence an increase in the volume.
It seems as if this can be generalized to any system as long as it's a linear function of its components, as it grows as a linear function of its components.
As long as the volume
Volume meaning the
the size
system, whatever you define the system.
Exactly. Yes. The size
It grows a linear function of its components.
linear. Actually, it can be. It doesn't have to be linear. It can be any homogeneous degree one function.
Oh, yeah. Yeah.
Okay. Yeah. Right. I gave you the. I mean, for simplicity, I presented a linear example.
Yeah.
But it could be any homogeneous degree one function, and the overall mathematical structure still comes out. So yes,
Could apply to even non-living systems also.
indeed.
Yeah.
Indeed. Pluto cells are non-living, and it applies to them, but it could also apply to human societies in some sense.
Yeah. So you mentioned that this applies to homogeneous systems, and you are assuming that the bacterial cell is a homogeneous system inside the cell wall.
Yes. So that's right. Okay. So indeed, the very fact that one is writing down that my state of the cell has no memory of where the molecules are inside the cell. It's only the total population that matters, and the change of the populations is governed only by the set of population, by the state which is governed by the population vector, if you like, and not where the molecules are located. So there are no spatial degrees of freedom. It's everything is only a function of time. In general, the concentration could have been a field, which depends upon space and time. Some concentrations are large somewhere and small somewhere, and so on. It's a model which ignores that complexity. It's a model.
So
It doesn't assume that homogeneous.
because the reason I was asking this was that when the bacterial cell E. coli divides, it has this FTSZ protein which sort of marks the middle of the cell where it will constrict and divide. So the fact that FTSZ comes to the center of a cell is because of a gradient that is from the two peripheries
Yes,
and sort of highest in the middle. So you are ignoring that.
absolutely.
And therefore,
Yes.
so if you, and you mentioned at the end, and this is like a very naive question, you mentioned at the end that there were like you could take multiple variables that can be accommodated in this equation. So did you try sort of looking at, because once the cell divide, it sort of reduces the volume to the original, and that's what it sort of the plots in the beginning were showing, right?
Yes.
It always came down on the multiple trajectories that you were showing, generations actually. So if you come down, it always comes down to the basic. One daughter cell will come down to the sort of the bare minimal and rise up, come down to the bare minimal, rise up. So essentially it's constricting, coming down to the original volume and then going up. So essentially this cycle that is going on seem to be dependent on some concentration of certain molecules that are determining what is the appropriate size at which to divide.
Exactly.
What is because it's dividing at the center, so obviously you're getting equal size daughter cells. Yes. So is that something that
Absolutely. So this model, so I've only talked about explaining the growth part of this, all those trajectories, right? Why is that growth exponential and why is it balanced? One has to put in more structure into the model to talk about division and growth and division. However, and it would seem, I mean, so we know, you're absolutely right that there is a spatial structure which is involved in division. FTS molecule, FTSZ is not everywhere in the cell. It's located in the center and it's pinching the thing. However, one can describe division in a poor man's way without having to make recourse to spatial degrees of freedom, okay? By simply saying that, let's say, you can say that when the volume reaches a certain critical threshold, the cell just divides into two cells. The volume just halves, okay? You can declare that. Okay. Here is actually a model in which we don't say that, but we say that there is another molecule Z, which is a protein, which is a timekeeper molecule. It could be the DNA A molecule. It could be the FTSZ molecule, actually. And when the number of this molecule reaches a certain threshold, then the cell divides. Without committing ourselves to the fact that that is actually going to be spatially localized. But if you simply make the assumption, we don't know, supposing you make the assumption that when a certain threshold in this number is reached, then the cell will divide. If you do that, then you can recover lots of experimental results about all those trajectories and cell-to-cell variation, because this is a stochastic process, and so many other things can be done. So while the real world has that spatial structure, okay, a lot can be achieved by toy models which effectively take into account that spatial structure without explicitly putting it in. Okay, and that's what the sort of art is right now, because of course you can always talk about those detailed models, but then you'll get lost in the theory. Okay, and to really get at the essence of it, that's what people are trying to do.
Hello. Yeah, here. So, nice talk. Just one question. So, in your, you're using this OD-based models. And so, basically, like the OD-based models are based on the assumption that your system is working in the thermodynamic limit, where the number of molecules is like tends to infinity, and also the volume tends to infinity. Right. So, but like, if we go to a more realistic kind of like model, like using stochastic kind of models, like, so do you get the same kind of results? Like,
Absolutely. Okay, so indeed, that's absolutely correct. That one is, the continuum model assumes large populations. Okay? But some of these populations can be small. And so you can look at the stochastic version of the same dynamics, which we are looking at the deterministic dynamics. You can look at the stochastic dynamics, in which the variables are not continuous numbers, but they are integers, non-negative integers, and populations can be 0, 1, 2, 3, 4. And each reaction has a probability rather than a reaction rate. Each reaction has a probability to happen. And you can do those kinds of things. And it is precisely those kinds of things that allow us to recover these stochastic effects. The cells are not going to be identical then. Okay. And so, however, balanced growth and exponential growth still come out. Okay. But stochastically. So, as you saw in those experimental results, the red curve that was growing exponentially had jumps. It was the only average that was exponential. Right? So, that is the feature that comes out. You see those fluctuations about the exponential growth, but the average turns out to be the exponential.
So, what is the model like you are using for the stochastic simulation?
So, what you say, you can use, for example, the Gillespie algorithm. I don't know if you're familiar with that. So, you have rate equations for the deterministic version, but the very terms that you have in the deterministic rate equations, you can relate to the probabilities of those reactions happening. So, at any given time, you have a state of the system, which is given by the number of all the molecules, which are now integers. But then there is a probability for a particular reaction happening, and you update the system by choosing each reaction according to its probability. And at any given time, a particular reaction happens, then you update the populations based on which molecules are consumed and which molecules are produced in that reaction. So that's how you construct a stochastic trajectory of the system.
Thank you.